Okay, in this lab, we're going to be focusing on metabolism, and we are just doing the fermentation tubes in exercise 8. We're going to skip over those oxidation reduction tubes, OF tubes. Okay, most bacteria are like our cells in that they can use various different carbohydrates as an energy source. Now, of course, most of them can use other kinds of macromolecules for energy as well, like proteins. We're going to talk about that a little bit. Other things like lipids, even DNA sometimes. But in this lab, we're going to focus on the use of carbohydrates for energy, and we'll see that there is a variability among bacteria as to which sugars they can use and which ones they can't. So we often look at this, especially in gram-negative organisms, as a way to identify organisms. Oh, this one can use sucrose and this one can't, for instance. Okay, so remember this term fermentation. We talked about this term a little bit before. Remember, fermentation is not the same thing as anaerobic respiration. Often we group those two things together because neither one of them require oxygen. But remember, anaerobic respiration typically goes through some kind of a Krebs cycle and an electron transport chain and some kind of inorganic ion, like maybe nitrate, is used as a final electron acceptor. In fermentation, something typically organic already there in the cell is used as a final electron acceptor, like pyruvate. Okay, and how can we tell which is going on in these tubes? Maybe there's some aerobic respiration going on as well. We can't tell for sure, but the end products of fermentation with bacteria are highly acidic. Okay, so we're going to test for a low pH there as evidence that fermentation has taken place. You will get acidic end products from anaerobic respiration or aerobic respiration, but not as much. All right, so when the pH dips pretty low, we can be pretty sure that fermentation's taking place. You know, it may be some respiration as well, but a lot of fermentation. Okay, so let's look at the media that we're going to be using. We often call this fermentation broth, or sometimes we call it purple broth, kind of for obvious reasons here. This is what the purple broth looks like before it's been incubated. Okay, so in these fermentation tubes, we have a 1% sterile sugar medium. Okay, and we have 4 different tubes. Now, they pretty much look alike, so make sure when you're picking these up in lab that you label them right away, which is which. So, like over here, this is the lactose, looks like maybe this is a sucrose, put an S there, mannose, and glucose. So, have some way to label those when you first pick them up because they won't look any different. Okay, again, one tube has glucose, one has lactose, one is sucrose, and one has mannitol, which is a reduced 6-carbon sugar. Okay, so that sugar is added to a kind of basal nutrient medium. Okay, we also need pH indicator in there. As I said before, we're looking for acidic end products as evidence that the organism has fermented the sugar. Okay, and this indicator we're going to be using is called bromcreosol purple. And with this pH indicator, we get a color change at about 5.2, so it's purple above that and yellow when it's more acidic than about 5.2. Okay, there's also some peptones in this medium, some short chains of amino acids, and we're going to get back to why that's important later. Also in the glucose tube, there is a durum tube. And I kind of hurriedly explained what that was when we did our nitrate reductase test. You can see here the durum tube. This is, again, a little inverted test tube in the larger test tube that tests for gas production. This is just one way that we can further characterize the fermentation pathway that an organism uses. Does it produce a gas as a byproduct of fermentation, or does it not? Okay, so convince yourself when you inoculate it, you can see that the media here is totally filling up the durum tube. So after you incubate, you're going to look to see if there's a gas bubble in there or not. And sometimes you have to look kind of carefully there. Sometimes it's really obvious. Some organisms are huge gas producers as a result of when they ferment. Okay, again, it depends what pathway the organism uses. Okay, so for a positive fermentation test, we're looking for a yellow color change. Okay, we're looking to see if the pH changes and also characterizing as whether a gas is produced. Okay, some students get this gas production a little bit mixed up. Of course, an organism is never going to produce a gas if it doesn't ferment. But if it does ferment, it may or may not produce a gas. All right, so we need to have that yellow color change as evidence there. Okay, one other thing, why do we only put a durum tube in the glucose tube? Well, it's a pain in the neck to put in there, but that's not the only reason. We assume that if an organism produces a gas when it uses glucose, that it would produce a gas for any other sugar that it fermented as well. Okay, so if positive for gas when glucose is fermented, will be positive for other fermented sugars. Okay, because basically the other sugars are typically broken down to glucose and then fermented, and typically one organism will use the same pathway no matter what sugar it's utilizing. Okay, so here's an example of what the tubes may look like after incubation. Usually we will incubate for up to 2 days at 35 degrees Celsius, and you can see here we've got a couple positives, whatever this sugar was is negative. It's not unusual to see some variability there. And if you look really closely, you can see a tiny gas bubble there in that positive tube as well. Okay, one other thing that I want to bring up in terms of reading these fermentation tubes that we have to be aware of. What can give us erroneous results? Well, one big thing we have to pay attention to here is incubation time. If we incubate these tubes for too long, say we leave them, forget about them, leave them in the incubator 3 days or 4 days, or say we inoculate the tubes too much. It's not something you're likely to do, but if you say inoculated those tubes with 4 or 5 loopfuls of your stock culture. And I didn't talk about inoculating here, but you're just going to inoculate them like you would a normal broth, either touching some growth from a plate or getting a loopful of your stock culture and swirling it around in there. Okay, but if you do either one of these two things, incubate them too long or inoculate with too many cells to begin with, what's going to happen is that your organism will basically eventually use up all the sugar. Okay, and what happens when that sugar nutrition source runs out? Well, it could be that the organism would die, but that's not likely to happen because remember I said most organisms are a little more flexible in that, and they can use other macromolecules for energy. So typically, the other macromolecule that's there and available to use for energy are proteins. Okay, so we have a phenomenon, we call this process alkaline reversion. Okay, so to begin with, an organism, again, is using sugars. Wait, let's back up a minute. To begin with, the medium is purple, right? It's neutral to start out with. And then when the organism starts using the sugars, or I should say fermenting the sugars, then we get a yellow color change due to acidic end products like lactic acid, or formic acid, or acetic acid, any of those things. But again, if you're incubating too long or for some reason the sugars get used up, then basically the organism will switch and start using proteins instead. Okay, the end products of protein utilization are not acidic. You're going to get things like ammonia as byproducts. Remember when we talked about phenylalanine deaminase? A deaminase cleaves off an amino group from a amino acid. So that's where we get things like ammonia as an end product. And now we're going to get the color change back to purple because the end products now are more alkaline. Okay, so originally we had some acidic end products. So if you're only looking at your tubes now, maybe after 3 or 4 days, and they're purple, that would give you a false negative because you totally missed this yellow color change over here because you weren't paying attention. Okay, so I just want you to be aware that that can happen. That won't happen. We'll take your tubes out after a day or two, and if they're purple, you're going to say, oh, the organism is not fermenting that sugar. If it's yellow, then yes, the organism is. Okay, but you just need to be aware that alkaline reversion can happen if you kind of are not paying attention.